you all for coming very much. Jonathan just gave me the thumbs up sign on the dot, so scientists are prompt, so thanks for being here. That's great. I do want to introduce Amanda Eckerman here in just a second, but I also just want to briefly, for those of you that haven't heard of the Continuum Scholars Program, just let me say just a few comments about that. Amanda is the final speaker in our Continuum Scholars Speaker Series that we had this semester, and that all kind of goes back to work that we did not quite a year ago. It was in mid-May with a group of 5 faculty that were all in their 3rd or 4th year here at Hope. So pre-tenure, but not brand new. And it's called Continuum because it follows Initium. And for those of you that are students, what the heck? But anyway, those are sort of faculty development programs that have been put in place. And so Initium, something that is for faculty that are brand new here, and then we thought Continuum would be something that could be still pre-tenure, but when it's been a few years. And it's an interdisciplinary program. It started, the 5 participants were Jared Ortiz from the religion department, Chad Carlson from kinesiology, and then Lindsay Root-Luna from psychology, Lena Chavez, who presented just a few weeks ago, and then now Amanda Eckerman. So it was a great interdisciplinary group, and it was really, really a lot of fun. Jonathan Haygood, who's in the back, and Andy McCoy, who will be here after his class finishes, and I kind of facilitated that group. So we met a few times during the summer, and then really the idea was to think a little bit about vocation and to think a little bit about how our faith impacts our scholarly work. So that was the program. We were very excited to have 6 new faculty that are going to participate in the program this May. So it's going to be great again. So as part of that program, each one of us, including the 3 of us who were facilitating, wrote kind of a non-traditional little biography paragraph. Not that, I mean, as you'll see from what I'm going to read that Amanda created, there are some of the same mile markers about where you went to school and so on, but it's just maybe a little bit more personal. So that will be kind of my introduction. These are her own words, but she's recrafted it to be in the 3rd person, okay? Because her father was a scientist, there was never any question that Amanda would be a scientist when she grew up. In high school, besides science, she loved creative writing. Like many teenage girls, she thought she could write really great poetry. But she knew that she wouldn't pursue writing as a career. In college, Amanda had a great experience with research in chemistry, but bad experiences looking for a church. Graduate school at University of Illinois at Champaign-Urbana fueled her love for making new molecules. There, the first church in Champaign, Illinois that she visited was filled with chemists, literally. She chose to be baptized by full immersion at the Hessel Park Christian Reformed Church, and a few years later, as a freshly minted PhD chemist with her expertise, the jobs available were well-paying, but frankly uninteresting. You'll hear a little bit about that today. Amanda decided to do a postdoc to do some more meaningful research. And during her postdoc at Northwestern University in Evanston, Amanda learned that creativity and good writing skills were vital to being a good scientist. After her postdoctoral fellowship term, she stayed on at Northwestern as a research assistant professor. When her daughter was born, she made the decision to leave Northwestern. And it was then that she discovered her devotion to undergraduate research. And of all the accomplishments she could write about in her applications for jobs, her favorite was that she was able to provide over 30 undergraduate students with a research experience at Northwestern. And she continues, some of you here, I'm sure, know Amanda well, either from the research lab or from courses that she's taught. So looking forward to kind of her reflections today, which are not, I think they're true stories, but then she remembers stories of truth from the lab. And hopefully she'll wrap up in time that we can have a few questions. Thank you, Amanda.
Well, this audience is a lot more chemists than I had anticipated. Sorry. When I tell people that I'm a chemist, the main reaction I get is, oh, I did not understand that at all in high school. And that always makes me really sad, because I really believe that inside everyone is a tiny little scientist. It might be easier to imagine if you first imagine yourself as a kid, but if you picture yourself with a white coat and a magnifying glass, right, you can picture yourself as a scientist. Kids are interested in the smallest of things, right? Things that we sort of overlook these days, right? Like snowflakes, right? Snowflakes. This is a fantastic website at Caltech where they study snowflakes. And dirt, right? My daughter is fascinated with dirt. This is another fantastic website. He has a PhD actually, but he takes pictures of microscopic things. So fantastic things. So inside all of us is a tiny scientist. Chemists are just people that never really outgrew that, right? So we continue to delight in nature and wonder at nature, the smallest things. We have an awe that grows with understanding, right? The more we understand a system, the more in awe we are, and the more respect we have for the complexity and elegance of nature. In particular, I like to have my students exposed to the definition of chemistry, right? In particular, we are interested in the composition, structure, properties, and reactions of matter, right? So we want to know what things are made of. We want to know what all the little molecules are, what shape they are. We want to know what they do. When we see a glass of water, for example, right, we don't see it as half full or half empty. We see H2O molecules hurtling through space, colliding with one another and exchanging protons. When we look outside these days, right, it's spring now, things are turning green. We can see the molecule that makes leaves green is very, very similar to the molecule that makes blood red. There are some differences here, but one of the main differences is this metal ion in the center. Right, we have iron, Fe, magnesium, Mg. So this is what makes blood red and leaves green. As you can see from this, chemists have their own language. We have symbols, we have pictures and structures, we have rules and grammar. When we develop this is to communicate our understanding of things with each other. When I see a glass of water, for example, or this bottle of water, I don't just see this. I also think about the structure of the molecules, and I think about the symbol and the name. So when I, as a chemist, think about water, I'm thinking about all of these things at the same time. And I know that that is not an easy thing for everyone. So I actually consider it to be a superpower, right? It's like having the ability to see the invisible, right? You guys don't see, well, with chemists here, you don't see these things in here, but I see it. And so it's kind of like being able to see things that are invisible. So another thing about chemists is that we love discovery. As part of my daily life in lab, like today, we love to mix and stir. We like to make new things. We like to assemble atoms in new ways. We like to discover molecules that haven't been characterized before. We assemble atoms in ways that never in the history of the universe have ever assembled in that way before. So that sounds a little bit maniacal, but and there are some maniacal chemists, but it's this discovery of the new that is so fascinating for chemists. But so we have this ability to see the invisible, but when we make something new, it remains invisible until we characterize it. So we do a lot of measurements and we use a lot of instruments and techniques and procedures to learn about our new things that we make. Okay, so chemists have these great superpowers. What do we want to use our superpower of being able to see the invisible and our love of discovery? What can we use this for? We work in a realm where we have many choices. In fact, everyone has these same choices. We have the choices of how to spend our time and how to use our gifts. So just like babies sort of awkwardly learn to use their hands, and when you're in your 20s, you sort of awkwardly learn to use your time. When you get older, you start to think about your gifts and how you can use them the best. So as part of the Continuum Scholars program, we talked about two words. We talked about vocation and occupation. Occupation you can think of as how you spend your time. So chemists can spend their time doing things like making more absorbent diapers, or a more durable paint, or a more refreshing toothpaste. These are things that chemists can do to use their time. But as a vocation, we must think about what we are called to do. What are your gifts? How can you use your gifts? As a Christian, some of these callings are easy. We are called to love God, we are called to love our neighbors, and we are called to be good stewards of our earth. So if we think of these sorts of callings, we can think as chemists, we should do things like develop drugs to fight disease. We should use our gifts to develop new ways to purify drinking water cheaply and efficiently. And we should work to deliver clean solar energy. So now I'm going to tell you a little bit about my vocational bio that was outlined in the introduction. So in high school, I was in a program called International Baccalaureate. And one of the most fascinating classes I took was called TOK. We called it TOK. It stood for Theory of Knowledge. And this class, students are asked to reflect on the nature of knowledge and think about the interconnectedness of knowledge and how we know what we claim to know. We discussed logic and fallacies. We watched a movie called Rashomon, which is arguably one of the best movies ever made. If you haven't seen it, go watch it now. It basically tells the tale, 4 tales from 4 different characters that all have the same encounter, and they all have a different story. So you're left with sort of the question of what really happened. We discussed the allegory of the cave, which is from Plato. We talked about the world as revealed by our senses is not really the true world at all, but only a shadow of it. So this is one of the classes I really loved. It was very challenging and very intellectually stimulating. Besides TOK, I took a lot of math and science classes, which I enjoyed very much, even though being a nerd was not cool then, like it is now. So this led me to go to school at a small school, college at a small school in Pasadena, Caltech. And so as a freshman, I was in a class, so I was going to study physics, and then I thought the chemistry was more fun. Chemists seem to have a lot more fun. So I changed majors to chemistry. So as a freshman, I took a class that really influenced me a lot. It was the class that started me on my path to research. This class was called the pizza class. And it was called the pizza class because it met every Thursday at lunchtime, and we had pizza. And all the students would eat pizza and fall asleep. Because the professors would come and talk about their research. And the goal was for the professors to recruit the undergraduates to work in the lab over the summer. But most of the time what they were talking about was, as freshmen it was over our heads and we were full of pizza and it was dark, and so we would fall asleep. It was a great class. So what happened was one day a professor came in and he said, "We're not turning the lights off this time. No one's going to fall asleep for my class. Leave the lights on." And he showed this picture. And I thought this was the most amazing thing that I had ever seen in my entire life. This is a protein. This is a ribbon structure of a protein. It's cytochrome C, which I know what these things mean now, but at the time I had no idea. So inside there's a heme, which is very similar to what I showed you for the hemoglobin. And this has been modified with the ruthenium complex. And they were studying electron transfer between this metal center and this metal center. But all I could think was this was the most amazing thing I'd ever seen, and I had to get involved with whatever this was. So I asked him, this was Harry Gray, so I asked him if I could do research for him that summer, and he found a spot for me. And my first project was actually to make a porphyrin that was one of those structures that I showed. And so I did column after column. The chemists here know what that means. I did column after column after column after column. And at the end of the summer, I isolated 0.3% yield of my porphyrin, which that was a great summer. It was a fantastic summer. I know that sounds a little bit sarcastic right now. One of the great stories that I love to tell about this is that I had isolated, I had my beautiful, it was beautiful green porphyrin, it was a solution, and I handed my round bottom, it was a flask or a glass container, to my lab mate to look at, and he spilled it all over the floor. This was everything I had worked all summer to make, and it was all over the floor. And he was like, "Don't worry, I will clean it up for you." So he did what is now known as a Kim wipe extraction. So he soaked it all up with paper towels, and then drained the liquid, and then did another column, and there I had my stuff back. And it was probably more pure than before he. So that's one of many great stories from that summer. So as I was doing all this, I was learning. Oh, and so the sort of the punchline of this story is that this molecule was one line, one sort of entry in one table in my graduate student's thesis. So this 10 weeks of work, this column after column after column was one entry in a table. So what I learned that summer is that there's a lot more than what you learn in the classroom. There's a lot more than what's in the textbook. There are new problems to solve every day. There's more and more new problems. And small pieces of data add up to make a big picture. So I worked in that lab for 2 years, 2 or 3 years. Different things. So the next summer, so everything went really well that summer. The next summer, nothing worked at all. So after that, I learned that it's okay if chemistry doesn't work. It's okay if things don't work the way you expect them to. But at the end of 4 years, or really 3 years, because at 3 years you have to kind of decide what you're going to do. What was I going to do with all these amazing things I learned in classes and in my research? And so I reflected a little bit on my faith, but I wasn't really sure yet. I felt like I hadn't learned everything. I felt like I really needed to learn more. I needed the excitement of a challenge. Being a technician, bachelor's of chemistry at the time, I felt like I was only going to really be able to get a bachelor, sort of a technician job, and I didn't really want to do that. So I went off to graduate school. I went to graduate school all ready to learn more things about chemistry and make even more new molecules. And I especially wanted to do something with iron. So this is iron here in the center. I had learned that iron does a lot of interesting chemistry. There are lots and lots of enzymes, proteins in nature that do reactions. They do transformations. They take smaller molecules and they put them together, or they cleave them apart. And so I really wanted to do something with iron. So I joined a group that lured me in and said that I would be making models of metalloenzymes with iron thiolates. So thiol is sulfur, so iron sulfur based enzymes. So I started working, and not too long after I had started working, a fellow graduate student, an older graduate student who had sort of maybe been assigned to look over me and help, insisted that we have a meeting with our advisor. So I went into the office and I sat down. They were already there. I sat down, and this is a little bit paraphrased. The other graduate student said, "She can't work on iron. I'm working on iron. She can't work on iron." And so I was like, what did I do? So my advisor said, "Fine, she can work on ruthenium." Okay, so here's ruthenium, for those of you that don't know, that don't have this memorized. Ruthenium is right underneath iron. The periodic table is organized so that elements that act the same way, that react the same way, have similar properties are sort of grouped together. So ruthenium being right under iron is similar to iron. A lot of the reactivity is the same, but it's not iron. And there are no enzymes in nature that use ruthenium. So I worked with ruthenium because that's what I was told to do. And I reacted a lot of different ruthenium complexes with different sources of sulfur, making different ruthenium sulfur clusters. And it was fun. I didn't really object because I knew that whatever I was going to do, I was going to learn a lot. And this is one of the things that I made, one of the molecules. My movie here. I was very proud of myself for making a movie. So there are 4 ruthenium centers, and the blue is the ruthenium, it's a tetrahedron, and each side is bridged by a sulfur, that's the gold. And then also on each ruthenium is a phosphorus. And there's more on here. I just wanted to zoom in sort of on this core structure. So this was very beautiful. This is a beautiful structure. There are very, very few other compounds like it in the literature. And I made it once, and then I spent a year trying to make it again. And the real reason, so this had a lot of promise. It was a very unusual structure. It looked like it could be reactive. This is a very unusual coordination geometry for ruthenium. Coordination geometry just means how many different things are around that ruthenium center. So the real problem was that we didn't have a way to see this. So as chemists, right, I said that we have this ability to see the invisible. Well, this was a black hole. There were no analytical handles for this. It was paramagnetic. There was nothing in the NMR spectrum. The mass spectrometry techniques that we had didn't work. Finally, in a moment of either genius or desperation, you can decide, I decided to use a technique that was not normal for an inorganic chemist. I used MALDI, matrix-assisted laser desorption ionization mass spectrometry. It was basically the last type of mass spec that there was in the lab to do. It was sort of a last-ditch effort. But it worked beautifully, and so I can now analyze my reactions and see if this was in there, and refine our procedures and purification procedures and things like that. So this is what I did for a year. So I made this, I made some other very similar, very symmetrical structures that were very beautiful. And at the end, I had, if you remember, all the way back now, I had wanted to make something that did something, right? I didn't want to just make molecules. It's cool to make pretty molecules, but I wanted them to do something. So we tried some catalysis, we tried some polymerization. None of it worked. These things are dead dogs. They don't do anything. So then it was time for me to graduate and get a job. And once again, the jobs that were available were for things like big oil. Okay, so the one thing that metal sulfides are actually industrially used for is a process called hydrodesulfurization. And so that's a big word, but it just means use hydrogen to take the sulfur out of the oil that comes out of the ground. So oil comes out, we do a lot of things to clean it, and then you can burn it or use it for other things. Okay, so according to things that I believe, this is not the worst thing that you can do with your life, right? This would be a good, being a good steward of the earth. But honestly, you could look at it that way. But I sort of felt like this kind of thing is sort of like eating an Oreo after you brush your teeth, right? Right? Like you're trying to clean something while you're still making a mess, right? So it didn't jive with me. So, and I really loved mixing and stirring, and at this point I really loved ruthenium, so after spending 5 years with it. So I went to Northwestern. I got a postdoc at Northwestern, and I was very excited about this project that I was brought on to do. The professor that hired me, Tom Mead, had founded a company that is now called Genmark. It's still in existence. It's gone through a couple of different owners and modifications, but this is exactly what I wanted to do. Right? This sounded totally amazing to me. They developed a handheld device. It's actually not handheld. It's about this big, but you have a chip that's about this big. I don't have one. It's like this big. It's about inch by an inch. That you can apply a sample from a patient, a blood sample, saliva, some sort of bodily fluid. You stick it in the machine, and it tells you what DNA is there, basically. So if you're looking for a particular gene, for example, when I made this figure a while ago now, in 2006 they were FDA approved for the detection of the cystic fibrosis gene. In 2008 they were FDA approved to detect the gene for some people have a genetic sensitivity to warfarin, which can be very bad. If you take this blood thinner and you have this reaction to it, it can be fatal. So it's good to know if you have that or not. Since then, they've been approved for things like a respiratory virus panel, so all different kinds of respiratory viruses, and some other things. So this was not the project that I was called to work on, but the way this worked is there is a single strand of DNA on this electrode, on one of these little pads here. And when the complementary strand binds, it has to be perfectly aligned. It has to be a perfect match, and then there's an electronic signal that can be measured. And it's slightly more complicated than that, but that's the gist of it. So the project that I was called in to work on, to use ruthenium for, was a protein electronic biosensor. So again, we have a chip. We have on these electrode pads, we have these anchors that have a specific capture ligand. And so this is just some sort of peptide or something that binds very specifically to a protein of interest. So the other company was detecting DNA. This is about detecting proteins. There are a lot more proteins in your blood and body than DNA. So this could potentially be a lot more specific. So my job was basically to do a proof of concept on this, to synthesize these electronically active ruthenium complexes with a binding ligand and an anchor, and do electrochemistry. So this was great. I had a lot of fun with this. I got to work with ruthenium, and incidentally, my coworker was doing the very similar sort of other half of this project, was working with iron. So after a couple of years of working on this, I had a couple of publications, and I thought, okay, postdocs, this is what you do after a PhD, is normally only 2 or 3 years. So I thought it was time for me to look for a job. So I went to some information sessions for big companies like Dow and Exxon and DuPont. And I remember meeting with a guy, a representative who had a PhD in chemistry from DuPont. I think he had probably graduated from Northwestern, I'm not sure. But he told me that he was in the fabrics division. And I thought, wow, okay, so what do you do with fabrics? Well, there is a great need for fabrics to be strong and durable and resistant, especially for things like restaurant napkins. And I thought, wow, that is absolutely not what I want to spend the rest of my life working on. So I decided to stay at Northwestern. Tom was happy to keep me on, and this is when I became a research assistant professor. So this is where the stories of truth are going to come in. I have been doing chemistry now for quite a while, and so I had learned that there are basically 3 flavors of results that you can get from an experiment. It's a little bit like going, these ice cream machines, right? You have the one flavor from one handle that you like. You're like, double chocolate or whatever. And then, so that's the kind of result that you like, right? that you want. You get what you expect. The data support your theory, right? So you've done an experiment. You have this picture in your mind of what should happen. So when you get this is cause to celebrate, right? But the second flavor, right? This is the second handle over here. This is going to be something like fat-free vanilla yogurt, something that you really don't want, right? This is going to be I was trying to think of what kind of ice cream flavor that nobody likes, but it's just got to be like the fat-free frozen yogurt or something, right? So these are the results that you don't expect, that do not support your theory. So then there's a lot of questions. Was the experiment performed correctly? Were the data collected appropriately? What can we learn from this experiment that we didn't know before? Can we modify the experiment and try again? Do we have to scrap the theory? Okay, and then there's the middle handle, right? So you can have what you want, you have what you don't want. But what does the middle handle do? Right. You get mixed results. Yeah. All right, so let me tell my story now. All right, so this is a project that a graduate student was working on. This is going to be the greatest thing for medical imaging. We were going to make an iron oxide nanoparticle, which is a great contrast agent for MRI. And to that, we were going to attach a gadolinium chelate, which is also a fantastic MRI contrast agent. These are actually two different kinds of contrast agent. These make dark spots and these make white spots in the MRI. So we were going to introduce this into a biological system. And when this got to where it was going, this disulfide bond was going to be cleaved in the reducing environment of a cell. And when they were cleaved, they would separate, and we would have the nanoparticles would show a dark image where they localized, and the gadolinium agents would be bright where they localized. Okay, and then so this is a brilliant scheme for a lot of reasons. Mixing two thiols to make a disulfide is like this easiest reaction in the world. Your grandmothers could do this reaction. This is not hard chemistry. Making these two things separately, that's different. But once we had these two, we mixed them together, and using our analytical abilities that we had, we could see that we had attached these. They were covalently bound. We could wash these, we could dialyze this, and the iron to gadolinium ratio stayed the same. So that means this is stuck. But then, so the other part of our scheme for this was that this should be reversible, right? We should be able to go back. That's what we wanted it to do in the cell, in the biological system. So we took our material and we added a reducing agent. We added 5 different reducing agents. We added thousands and thousands of equivalents of reducing agent. This doesn't come off, ever. Actually, I'm exaggerating a little bit. Some of it came off, but not nearly all of it. So we could have explained this away. We could have published it. We could have explained this away as kinetic effects. This is on a surface. This is a giant. This is not to scale, right? This is a giant nanoparticle. Maybe we have kinetic effects. But the advice so we didn't understand this. We were flummoxed. I have to use that word. Flummoxed. So talking to Tom about this, he said that in science, you have to always make sure that you are pursuing the truth. You must never allow yourself to be pursued by the truth. So basically that just means when you're seeking the complete understanding of something, you can't allow yourself to stop questioning, right? You can't, when you think that you have found the truth and you think you know what's happening, that is when you have to be the most vigilant. You have to continue to question, even when the results of the experiment are exactly as you predict, right? So the first part of this went exactly as planned. There's no reason for us to think that that disulfide bond was not forming. So when the results support your theory, maybe we are closer to the truth, but maybe our theory, maybe our whole picture. So this is what I like to call cartoon chemistry, right? These are all cartoons. We don't really know what the surface of the nanoparticle looks like. This we can characterize pretty well. This is a molecule. This gets a lot more fuzzy. So the bottom line here is that if you want to keep the truth off of your tail, you have to keep it front and center. The correct reaction to any result from any experiment is really, interesting. What else can we do? As a chemist, it's really easy to get to the point where you really feel like you can predict everything. You can predict a lot. You know a lot. You can really control what's going on in your reaction flask. And that's exactly when it becomes very easy to overlook, to unintentionally overlook or ignore data that doesn't fit into your theory. So we wound up not publishing this at all. There were some other interesting things that happened that I'd be happy to tell you about later if you were interested. But I'm talking long here. And incidentally, this is when creativity comes into science. So this is where you want to have conversations and get a different perspective. Okay, so I'm going to talk quickly about work at Hope. I finally got to the point where I could work on iron, but I'm not working on iron, I'm working on ruthenium. Using ruthenium still. So things that I want to do here at Hope. I want to address issues that I think are going to become more and more relevant. So in our aging population, these are some stats. By 2050, the amount of folks in the population that are over 65 will double. And these are the ranking of causes of death. So heart disease, cancer, stroke. Most of these are preventable. Most of these are well enough understood that they are preventable. You don't have to get heart disease. If you eat right, exercise, no problem. Right? There's very, very, very little that you can do or that you cannot do to prevent that. Cancer, there are a lot of things that you can do to prevent cancer. But there's one down here that I'm especially interested in, and that's Alzheimer's. And I'll talk about that in a minute. First, I'm going to talk shortly about cancer in America. These are some statistics that my students dug up for me, which I checked. Where they're going to come from. So in 2015, there was predicted to be 1.6 million new cases, which is huge. It's a huge number of new cases. 1 of every 4 deaths is associated with cancer. Right? So that's from the previous slide. A third of these cases are related to things that you can prevent and avoid. Right? So we could all just go for a walk for 20 minutes a day. Not even 20 minutes. Right? You could go and walk for 5 or 10 minutes a day. And you could eat your veggies. Right? Eat your veggies. So this is Cisplatin. This is a hallmark intermediate complex. This is used for cancer treatment. It binds the DNA and makes the DNA shape change, and then the cell dies. There's a lot more that happens than that, but that's it in a nutshell. Unfortunately, this is a small, very non-specific molecule. So one of the reasons that it makes patients very sick is because it's non-specific, and it kills lots of healthy cells as well. Now, there was a recent paper that showed this ruthenium complex, that it binds to cellular chromatin. You can't see it very well here, but this is a crystal structure, actually, of this complex binding to the protein part, and it has ruthenium. This is very exciting for me. And what's fabulous about this compound is that it actually inhibits metastases. So in cancer, most of the most of the deaths are from metastatic cancer, not the primary tumor. Metastatic cancer is what we need to go after. If we can keep cancer from metastasizing, we have a better chance of surviving. So, wrapped up with a couple of other things I know about cancer. We decided to, these are some of my goals. So I want to make an anti-proliferative ruthenium complex, so that is an anti-metastatic ruthenium complex that has improved cellular uptake. So how can we get more of this or something like this into the cell? And this is well known. This binds to DNA. There's a crystal structure. This is all very well understood. How ruthenium drugs affect cells is much less understood. This is the second part of the goal. So Lindsay, a student in my group, has been working on making these sorts of complexes where we have a ruthenium with a glucose group. So my idea here is that glucose, which cancer loves sugar, we'll take this up more, and then this will be more effective. This is a collaboration with the Ludden group, Dr. Ludden and Luke. We're working on these cells. So this is, these are some breast cancer cells, and this is after treatment with this complex. So you can see that there are fewer of them, so many of them have died. There are some other differences too that we've quantitated, but this is a very promising start, I feel, for this project. And this is exactly what I want to be doing. I'm so excited. All right, so now I mentioned this other disease here at the bottom, Alzheimer's. So it doesn't account for too many of the deaths, but there are some interesting statistics here. Alzheimer's, sixth leading cause of death in the US. 1 in 3 seniors dies with Alzheimer's or some other form of dementia. So maybe they don't die of this, but they have it. And what's most depressing about this disease is that currently there's no cure. There is no way of preventing Alzheimer's. And there's no way of slowing it down. So we see all these things, do Sudoku, and these are sort of good things to do, obviously, but there's not really preventing or slowing these diseases down. There's no single test, right? You can't get a biopsy. Brain, it's hard. So you have to you have to have a very skilled doctor. You can't, it's a difficult multi-step process. Medical history, and there's some imaging that can be done. But in general, this is one of the worst, I think, challenges, the most serious challenges facing our aging population. Okay, so let me just talk quickly about what's going on in the brain. There's a protein called A-beta, and this is a naughty little protein. It does all sorts of things that it's not supposed to. It makes these oligomers, small ones, larger ones, different sorts of shapes. It makes these fibrils, and some of these bind to neurons and are toxic. Okay, so it took scientists a long time to figure out. For a long time, it was thought that these fibrils were bad, right? It seems like a bad thing to have solid precipitating out in your brain. But actually, it's these soluble oligomers. So this is not really a chemical reaction scheme that we're looking at here, but if you can picture these as sort of double-headed arrows, right? We have the monomer, everything is in equilibrium. So if we can influence this equilibrium, so if we can influence the formation of these away from the soluble species, the soluble toxic species, maybe we can make more of these that are sort of benign, make more of this monomer that can be cleared away. That would be helpful. At least if we could understand different factors that make these form, that would be useful information. So this is the whole molecule. This is the sort of the part we're interested in. These are the sort of the hydrophobic areas. And so we have designed, this is Morgan's work, we've designed this large ruthenium cluster. So this is easy to make, easy to put these hydrophobic ligands on. This is beautiful data that she has acquired. So this is not terribly difficult chemistry, and it's a way. So the idea here is that we can make a large molecule that has hydrophobic groups. Maybe we can template the formation of different shapes of oligomers. Maybe we can template something different to happen that is not toxic. So she's done. This is beautiful data here. This shows that we have this in solution. This is a mass spec. This shows all the different isotopes, a combination of different isotopes. And this shows all the different electron transfer events that this ruthenium complex can do. And we also got a crystal structure, which is beautiful. So, sort of in summary, there are summary points. Oh yeah, that came up. All right, so the first thing that I hope that you take away from this is that when scientists study something, we hope that it will help us understand something that is more complex. Experiments are building blocks to greater knowledge, but we have to continue to question everything. So even if you're not a scientist, you can continue to question everything. How do you know what you think you know? And as you can see here, we have here at Hope a great philosophy department, and there are emergency philosophers on hand to administer aid in case of an existential crisis. I think you can take that question pretty far. Why are you here? What are you here for? Why are we here on Earth? I believe that scientists are scientists because we're human. We are curious creatures, and there are all these fantastic puzzles here for us to figure out. From a theological perspective, one can consider that theories come from the human mind and therefore may be inherently flawed. Or, since if you believe, like I do, that God created the human mind, we should be able to understand everything using pure reason. So this is a gift that we've been given. We may have to accept that we will never attain complete understanding, and that we may be on a search for truth, knowing full well that we will never actually find it. So, spend some time reflecting on why you're here, as I did at different points throughout my career. Think also about where you were and where you're headed. And this kind of reflection is a human thing to do. And think about this idea that vocation is not necessarily your occupation, and that it's perfectly fine to make restaurant napkins. Thank you. I would be very happy to take any questions. Sorry, I didn't have more time. When I practiced, it was only like 25 minutes. I'm
concerned about that. Are you pulling those students out?
No, I'm capturing them. I have captured them. I have captured the best undergraduate researchers at Hope. They're in my lab under the beaker. Yes?
I'll buy an answer to this question. Why is it okay to make napkins like that?
Why is it okay?
Yeah, I agree, but I'd be curious to hear you. Why is it not okay for you, and why is it okay for others?
Why is it okay for someone else? Well, I'm driven by this desire to be intellectually stimulated every day. Someone else may be driven to have a high-paying job where they can devote more time and energy and resources to their family and to their church or to charity functions. Maybe making restaurant napkins is a stepping stone on a career path where they will eventually make smart baby seats, right? So if we have to have car seats, maybe that could be a longer-term goal for someone in that career. Yes?
What called you to Hope College?
What called me to Hope? Well, when I visited and I saw the labs bustling with undergraduate researchers, I thought, wow, this place is just like Caltech. I want to come here. I want to be a part of this. I was really impressed with the research and the dedication to undergraduate research here. Yeah.
So, how do you inspire students to do this? Because many of us here teach, and you get students who don't question things. They just say, I want to know the answer. I want to be done. And they go make those napkins. It's like it's frustrating, and you have to have a high tolerance of frustration, because clearly a lot of the students aren't curious, especially in the sciences. We have a lot of students who are taking a science course for simply a reason to check a box on an application, and they could be not curious at all. How do you deal with that? Because it's run across multiple times.
Yes, I'm still learning how to deal with this. I would say that when you're a college student, you think you have all the time in the world, and then one day you wake up and a lot of time has gone by. And encouraging this reflection on a frequent basis, I think, is something that we should do. You can wake up and you go to college, check. You get a job, check. You get married, check. You get a family, check. You die, check. Is that really what we should ask our students? What do you want people to say about you at your funeral? That's something we should all think about. Did you just go through life, or did you think about why you're here? At least once in your life, you should think about that.
Oh, Jonathan's got one more.
You've spoken about wanting to be a writer or a poet when you were younger. How has that shaped what you're doing now?
Well, I mentioned it briefly. I think being creative is a really important part of being a scientist. When you get all these results that don't make sense, you don't know what's going on, you really need to be able to think about it from a different angle. You have to come up with some sort of creative new approach. Writing is very important to being a scientist, right? You have to be able to communicate your findings, communicate your thoughts, communicate your theories to other scientists. And really the only way you get good at writing is to do lots of it. And have someone read it and tell you, wow, that's really, I don't understand that doesn't make any sense, grammar is awful. And you go through that a lot of times, and you get better at it, and you find eventually that you don't really know what it is you're thinking until you write it down. Writing and thought are very, very intertwined. So that's how I continue that part of myself.